So another spicy problem here, 15.28, and this is I like this problem because I like shows like Star Trek and Firefly and Battlestar Galactica and what is my Adventure Time? Yeah, that's my new favorite. Anyways, okay, among betazoids in the world of Star Trek, Farscape also. I love Farscape. Okay, so among betazoids in the world of Star Trek, the ability to read minds is under the control of a gene called Mind Reader. Okay, that is pretty cool. So, ability to read minds. Gene called Mind Reader, an appropriately named gene. So betazoids can read minds. I'm not familiar with this episode of Star Trek. Actually, haven't seen that many episodes. I like the new Star Trek with the guy from Quantum Leap. Only lasted 4 seasons. I think it should have gone much longer. So, okay, so Mind Reader, abbreviated MR. Most betazoids can read minds, but rare recessive mutations in the MR gene result in 2 alternative phenotypes: delayed receivers and insensitives. Okay, that's going to be a huge disadvantage to individuals who, man, if you're in a society where everyone could read minds except for you, that wouldn't be good. Okay, delayed receivers and insensitives. Well, maybe it would be good. Yeah, I think that probably would be good. Or there could be advantages there. So delayed receivers have some mind reading ability, but perform the task much more slowly than normal betazoids. Insensitives cannot read minds at all. Betazoid genes do not have introns. Wow, that makes them really easy to study, so great. So the gene only contains coding DNA. Mind Reader is 3,332 nucleotides long, and betazoids use a 4-letter genetic code. Okay, we use 4 letters. No, we use 3 letters. They use 4 letters. Okay, the following table shows some data from 5 unrelated MR mutations. Okay, let me copy and paste that table in here. Okay, I'm resuming the recording here. Let's see, can I copy and paste that in? Yes, I can. Okay, we have our table. So the following table shows some data from 5 unrelated MR mutations. Okay, so we have MR1 through MR5. And what are we going to do with these now? For each mutation, provide a plausible explanation for why it gives rise to its associated phenotype and not to the other phenotype. For example, hypothesize why the MR1 nonsense mutation in codon 829 gives rise to the milder delayed receiver phenotype rather than the more severe insensitive phenotype. Okay, cool, let's take a look. Got my pen here. No, I don't. Okay, now I got my pen. Okay, so we're looking at MR1, and there's a nonsense mutation in codon 829. Okay, so that is interesting. Okay, first of all, let's look at how many codons are here in this 3,332 nucleotide coding region. It is, yes, okay. Okay, so we need to divide this by 4, right? 3,332 divided by 4. Well, let's see if I can do this by hand. I think this is looking good so far. I think I did it. Wow. Long division. I think that's called long division. I'm not really sure. So 8,833 codons. 833 codons of 4-letter codons are present in the mind reader gene needed to make that 3,332 3,332 bases are required to make this, are divided up into 833 codons. Okay, plus maybe a stop codon. I don't know if we included that. So it's okay, about 833 codons in this mind reader gene. So it turns out, so what is a nonsense mutation? So remember, nonsense is going from a amino acid coding codon, so a codon that codes for an amino acid, to a stop codon. So betazoids have stop codons; they must, just like we do. And this MR1 mutation has taken a codon that codes for an amino acid, and it turned it into a stop codon. And it's at position 829, so it's at the very end of the gene, only 2 or 3 or 4 codons away from the end. So that would kind of make sense that it didn't lose too much of the protein. So okay, so it is it causes the less severe delayed receiver phenotype. Okay, that makes sense, right? You have most of the protein there. You only lost a little bit off the C-terminal end there, the very end of the protein. So that makes sense why that would be a delayed receiver and not an insensitive phenotype. So that makes sense why MR1 would cause the less sensitive, less severe phenotype with respect to mind reading. Okay, let's take a look at MR2 now. MR2 is a missense mutation in codon 52. So what is missense? Missense. Do we remember what that means? This means where you have one amino acid. Let's say it codes for alanine. You have a codon that codes for alanine. And you mutate that codon, and now it codes for something different. Okay, let's say phenylalanine. Okay. So apparently, mutating this codon changes one amino acid, and we changed it in such a way that it didn't affect the protein too much. Still a delayed, only affected it a little bit, and so the protein still functions. The function is not completely lost. So it's not a null mutation or anything. So this is why we have the less severe phenotype, the delayed receiver phenotype. We've changed the amino acid, but it hasn't interfered too much with the functioning of the protein. It's not ideal, but we haven't completely lost function there. Okay, that makes sense. When you just have a missense mutation, you just change one amino acid. Depending on how important that amino acid is to the function of the protein, it may have no effect at all. It may just influence the function of the protein a little bit, or it could completely eliminate the function of the protein. In this case, it looks like it just interfered with the function a little bit. So let's take a look at MR3. So MR3 involves deletion of nucleotides 83 to 150. Okay, well, that's a total deletion of those, and we're still a delayed receiver. Okay, so something happened here. Well, apparently this protein is still functioning slightly, so this didn't have a major effect on the function of the protein. Well, I think one thing we need to consider is how long is this region here. So 150 minus 83, and let's see if I can do this by hand. This is going to be 10 here. Carry the 1. I don't actually know how that works, so I'm forgetting. Okay, here we go. 67. Okay, that looks right. So we deleted 67, and this is where we are going to run into this complicated situation where we're deleting everything between 83 and 150. So 82, position 82 is here, position 83 is next. Okay, then we're going all the way up to position 150. The next one's 151. So we're deleting all of these bases right here. So what's the best way to explain this? This is actually, I did I showed you a little math here, did the subtraction. This is actually not 67 bases. This is actually 68 bases, because 150 minus 67 gives us 83, right? But we're actually losing 83 also. So let me see, I'll show you this again. 150 subtract 67, we're going to end up, that's a 1 there, 15, hold on a second, 10, that's a 4, okay, that makes sense. 83, okay, but we're actually losing 83 also, right? Okay, so we're actually losing 68 bases here. Now 68 divided by 4, what is that? Hopefully that's a nice round number. Let's see, 68 divided by 4 is 17. So we deleted 17 codons exactly when we removed positions 83 to 150. So we deleted 17 codons exactly. So if this is our amino acid chain, and they're going to be, how many? We counted 833 amino acids. And okay, so let's say these ones right here, 82 divided by 4, what is 82? No, 80 divided by, what is going on here? Okay, so the easiest way to explain this is that, okay, we are losing 17 amino acids from near the beginning of the protein. And apparently losing these 17 amino acids does not have that much of an effect on the protein. And that can happen, right? So we took 68 bases, removed them, and it didn't have that much of an effect on the function of the protein. It did affect it a little bit, but the protein is somewhat functional. And okay, so it turns out that those amino acids were not critical for the function of the protein. Okay, so that makes sense. The reason I went through this complex math here is because if we didn't lose a certain number of bases that just cleanly cut out 17 codons, we could have caused a frame shift, and the frame shift would have a major effect on the protein, especially if it happened at the beginning of the gene. And we're going to see that with MR5. As you can see, I'm looking at it right now, I'm guessing that's what we're going to see there. So let's take a look at MR4 and MR5. These are mutations that were severe mutations, and they led to the insensitive phenotype. So MR4 is a missense mutation in MR4, missense mutation in codon 192. Okay, so we're comparing this to MR2, which was also a missense mutation in codon 52. Now, MR2 led to a delayed receiver, while MR4 led to an insensitive phenotype. So how can this happen? They're both missense mutations. Well, it just turns out that, okay, so missense mutation, you have a codon for one amino acid 1 we'll call it, okay, you have a mutation, you change the codon to one that codes for amino acid 2, a different amino acid. And it turned out in position in codon 52, so the 52nd amino acid seems to have been less important. So less important than the codon at position 192. So, I mean, that's one possibility. So position 52 is less important than 192. 192 is more important. The amino acid there, the specific amino acid there, is critical to the function of the protein, and changing it completely eliminates the function of the protein. So that's one possibility. That's one possibility to explain the difference between MR2 and MR4. Another possibility is that the type of missense mutation for MR2, we changed the amino acid to something that's, kind of similar, right? So alanine, for instance. Let's say we were in alanine. Now, alanine amino acid has, a CH3 side chain. So we can change alanine to glycine, and that's a missense mutation, and glycine has just a hydrogen side chain. So these are very similar amino acids. This would be considered a missense mutation, but the amino acid structure is similar, and so when you're going from one amino acid to another amino acid, if they're relatively similar amino acids, they're less likely to have an effect on the function of the protein. But we already came up with an explanation, so we won't worry about the second one. So our explanation we went with was position 52 is less important than 192, and therefore it's less affected by changes in the amino acids at that position. So let's take a look at MR5. So MR5 is going to be our contrast to MR3. So MR3 was the deletion of nucleotides, 68 nucleotides between for the delayed receiver, leading to a delayed receiver phenotype. And MR5 is deletion of nucleotides 83 to 93. Okay, so a much smaller deletion. So why did a much smaller deletion lead to an insensitive phenotype? That doesn't, on the surface, that doesn't make much sense, right? You would expect a smaller deletion to have less of an effect on the function of the protein. But let's see. So if we have, okay, we're looking at a protein or a sequence here. We go from 1 to 82, and then the next position is 83, and then we'll go up to 93 here. The next position is 94, and that's going all the way up to 3332, I believe, right? Okay, so those are just the positions of the, let's say, RNA molecules or nucleotides in this RNA or DNA, or in the DNA. These are just positions in the gene. So, and we are deleting now 83 all the way to 93. We're taking these out of the gene. So the deletion took out 83 to 93. And so again, if we just did the subtraction just like this, well, we'd have be losing 10 nucleotides here. But in reality, we're moving out, we're taking out 83 too. So 93 minus 83 is 10. 93 minus 10 is 83. Okay, that makes sense, but we're also losing 83. So we're actually taking out 11 nucleotides. And we can actually count this if we want. So we're going to take out 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, and 93. So now I can take my different color out here. Let's go with gray. Okay, let's count these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, we're cutting out 11 nucleotides. So there's some tricky math when you're working with DNA sequences and you're cutting out bases of how many you're actually losing. It's usually always one more than you think it is. And you can see why there with the explanation I just drew out for us, or diagrammed for us. Okay, so why does losing these 11 nucleotides have such a major effect on the function of this protein, while losing 68 nucleotides for MR3 had less of an effect. So losing 11 nucleotides cuts out, it doesn't cut out two codons cleanly or 3 codons cleanly, right? So 2 codons would be 8 nucleotides. 3 codons would be 12 nucleotides. As a result, we are causing a frame shift here. So what that means is the reading frame of the gene here, the mind reader gene, is going to be different after every position, after position 82, or somewhere around here, somewhere around position 82. As a result of this deletion here, every codon is going to be read differently by the translation machinery, right? So we can do an example of a frame shift right here. So here's a frameshift mutation, and let's assume this. We'll just use the betazoid genetic code with a 4 codon, 4 base codon. Okay, so I'm just going to put a bunch of letters down here, and I'm going to divide these up into codons. So codon 1, codon 2, codon 3, codon 4, codon 5, and so on and so forth. Now I'm going to cause a frame shift by deleting, not 4, I'm going to delete all 5 of these guys right here. Okay, so we have a deletion here that is deleting 5 different nucleotides. So I'm just going to put this guy over here now. C-A-U-G, just like that. Okay, now let's divide them up into 4 codons of 4. Okay, so this guy right here, this is codons the same. Okay, and then we have AGCA right here, and this codon right here used to be GAGC, now it's AGCA. So this codon, this new codon we formed, does not match either this one or that one. And let's see this one, GCGC. Well, this GCGC used to be, right here, used to be part of 2 different codons. So it codes for a completely different amino acid that wasn't even in the original protein. And if we looked at all the codons downstream of this codon, these codons right here, we would see that almost all of them are going to be different than what the original protein coded for. So frameshift mutations like this have major effects on the amino acids of the protein. So, and if frameshifts occur very early in the protein, like they did in this case, then you can change all the amino acids downstream of the frameshift mutation, and the protein is likely to be completely non-functional. So frameshift mutations are way more are more likely to cause complete loss of function of the protein than, let's say, a similar deletion where you're cleanly cutting out just the bases for a specific number of codons. So in the case of betazoids, it would be any deletion or insertion that occurs in a multiple of 4 would be less severe than any deletion or insertion that occurs in any other multiple. For us, it's multiples of 3 are less severe than any other combination of insertions and deletions.